Well, thanks very much, Shrug, and to the organizers for getting this together. It's a real pleasure to be here. So I've been asked to talk about metabolomics to characterize exposure. And this is sort of leading in or picking up from where Steve and Dave and Shrug have left off. And again, perhaps picking up where Steve brought up a couple of, I think, really important points. And this is just an example of lifetime risk for the 12 most common diseases in North America. And this is just a list of what's typically provided in many repositories or databases. And everyone in this room can expect over their lifetime to develop either a cold or the flu. 92% of us will develop cavities or tooth decay. 90% of us over our lifetime will develop hypertension. And you can go down the list. And these are all the things that essentially will strike at least some of us or all of us at some point in our lives. And what I've marked in blue on the sides are essentially the causes of these very, very common disorders. And whether it's colds or flu, it's viruses or bacteria. Tooth decay, it's what we eat in combination with the microflora in our mouths. Hypertension, it's very clearly an environmental condition. It's how much salt or fat or how much smoking. Same thing with heart disease and atherosclerosis. And you can go down, and every single one of these very common disorders, chronic or non-chronic diseases, can be attributed quite clearly to something that we're exposed to. The one exception at the bottom, number 12, is Alzheimer's, which is one where we have no clear understanding of what the origin and etiology is, but I think more and more people are realizing it might be sort of a type 3 diabetes of the brain, so it too may also have a clear environmental cause. If we look a little further and look at exposures and causes of death, and this is what we sometimes call the real cause of death. This is data that's been compiled from a whole bunch of different sources and picks up from a paper that was published in the mid-90s titled The Real Causes of Death, but updated obviously over the last 20 years. What I've listed here are things that, again, we can be exposed to. So people still die from asthma, which are caused by allergens. People die from drowning. Anemia, UV radiation, which causes a variety of skin cancers. People are still dying from asbestos exposures, even though it's been largely removed. Ionizing radiation from X-rays, CT scans, radon exposure in your basements, vitamin D deficiency as it relates to early death, and you can go down the list. And these are tabulated, and these aren't just made-up numbers. These are from a number of studies that I've compiled and updated with changes to population. You add it all up, and 2.2 million deaths out of the 2.4 million nonviolent deaths can be attributed to exposures. So that's more than 90%. And I think it reiterates a message that Steve was bringing up as well, is that the estimation, if you want, of the genetic causes of death or disease is perhaps wildly overblown. We were just doing another one that still hasn't been published, but counting up the other way in terms of the genetic cancers. And Steve mentioned a death rate of around 3.5% from the populational attributable fraction. Counting the other way, we came up with a number of 2.3% in terms of genetically caused cancers. So again, the numbers keep on, no matter which way we cut it, arriving at a number where far more than 90% of the causes of death and disease our environment, and much less than 5% is genetic. So this is the point that both Steve and I think others are starting to make. Most common and most complex or chronic diseases have an environmental origin. The numbers, as I've just shown, both in the US and Canada and many industrial countries, are really due to some kind of environmental exposure, and that can be either an excess or a deficiency. And fundamentally what this workshop and what the discussion over the next little while will be about is how can we measure those environmental exposures and those consequences. So I will posit that the answer is through metabolomics, not exclusively, but it is one approach that allows us to do a lot of the things that we'd like to do or have been wanting to do for many years. So to think of the metabolome, it's something that sort of caps off this pyramid of life, where the genome codes for the proteome in turn manipulates or alters the metabolome. And I've drawn this little bird or canary at the top of this pyramid to really indicate that metabolites are the canaries of the genome. So a small change in the genome, perturbation in the epigenome, or in the proteome is amplified to the point where it's easily detected. The other thing that I wanted to point out with this pyramid is, in fact, that what influences them in particular. And yes, the environment can weakly influence the genome, but it's intrinsically supposed to be stable over 80 or 90 years. What is influenced tremendously by the environment is the metabolome, because that's changing, and it's changing as you speak or think. It's also changing with the fact that you've just eaten breakfast. That's seriously altered your metabolome. It hopefully hasn't altered your genome. Same sort of thing as physiological influence. Largely every cell in your body is supposed to have the same genome, but every organ is largely designed to have a different metabolome. And we tend to forget this, and that our bodies are collections of organs and containers, if you want, each developed and specialized to alter, store, or manipulate metabolites in a very specific way. So physiological influence is also something that's encoded in the metabolome much more than perhaps the genome or proteome. So we've heard a couple of definitions of the metabolome. Again, it's really just the collection of small molecules found in a biosample. And typically it includes both endogenous and exogenous compounds. And the fact that it includes exogenous compounds makes it relevant to the exposome. So this is a picture that Augustin Scalbert has drawn and illustrated, and Stephen has mentioned Augustin a few times. The point is that what we have in our metabolome, there's both an endogenous one, the things that our bodies create, assemble. This allows us to do anabolism and catabolism. There's the stuff that we eat, that's the food metabolome. And then there's the things that we're exposed to, whether it's pollution or drugs. All of those things that are external actually are processed by us at some level. So digestion may process them, microbes may process them, other tissues may process them. So they alter. So there's the unaltered exposome and then the altered exposome that comes as a consequence of the action of enzymes and microbes. So when we measure exposures, and again, Stephen brought this up, which is there's three different views. There's the epidemiologist view, which is largely based on questionnaires where we ask people. And in that respect, the epidemiological view is largely indirect or categorical. What we want to do is elevate the game, if you want, and bring in something that's perhaps a little more precise. And this is where it's either the molecular epidemiologist view or the exposure scientist view. So the molecular epidemiologist would want to know about the biomarkers inside of you. So this relates, as Steve called, the blood exposome. And then the exposure scientist view, more an environmental scientist, wants to measure the external levels from air or water or dermal contact. Ultimately, what metabolomics can do is actually get answers to both. It can measure both inside and outside because, in fact, and we call it environmental metabolomics for exposure science and just regular metabolomics for the molecular epidemiologist's viewpoint. But the tools that you use are largely the same. The techniques are largely the same. So if we're looking at sort of the internal metabolome, this is a process that's typically followed by many people doing metabolomics now. So we'll either take a biological sample, a tissue sample, and because we like to do analytical chemistry, we prefer liquids, so we'll usually extract or homogenize those tissues. Sometimes we can save ourselves a few steps and we can just take biofluids. So blood and urine or saliva are often the favorite fluids that we tend to work with. Those fluids then are put into analytical chemistry equipment. So it's GCMS, LCMS, NMR. These are all tools that have been around for 50 or more years. And in fact, the techniques are not that different. Where the fundamental change has occurred is the processing and the analytics, if you want. It's this last step, which uses a combination of software and databases, machine learning, computers, to interpret the spectra that are measured in these instruments. Because now we're not measuring pure compounds, we're measuring mixtures. And mixture analysis is very tough. And it's only been in the last few years that, in fact, that's become possible in a sort of routine way. So that's the essence of metabolomics. To do the exposome, if you want the internal exposures, we would track what people have been exposed to. Things get processed in the body. We measure the or collect the biofluids and run through the same process. So one might have been looking at more endogenous metabolites. This way we're trying to track the exogenous/transformed chemicals that are in the body. And again, same pieces of equipment, same analytical tools. Same thing if we wanted to do environmental metabolomics. In this case, we can go around and sample foods. We can sample the air. We can sample the water. We can sample sentinel organisms to track what's out there and use exactly the same tools, exactly the same equipment, and measure these mixtures and quantify and characterize them. So in that regard, as I say, the concepts behind metabolomics, this multi-integrated chemical measurement process, can be used to look at all three components of exposures. As I mentioned, the technologies that we used in or use in metabolomics are diverse. There's chromatographic methods, HPLC, UPLC, capillary electrophoresis, microfluidics. Mass spectrometry is used extensively, very high resolution mass spectrometers. Also use ICP-MS to measure metals. NMR is also a very powerful technique, especially to characterize unknowns or previously uncharacterized compounds. Gas chromatography, also widely used, and even infrared spectroscopy. So these are all employed, and in fact, to do a comprehensive metabolomics study, often all of these pieces of equipment have to be used. That makes it a very complex kind of study. So this is something that, again, has been brought up, that it is difficult, requires people with multiple PhDs, and time-consuming. Different technologies used in metabolomics have different levels of sensitivity. The least sensitive technique is NMR. The most sensitive technique is LCMS. In between are gas chromatography. Now you could say, why don't we all do or just exclusively do LCMS? The reason is that as we get down to the picomolar and femtomolar sensitivity, many of the peaks that we see are simply not identifiable. So we're looking at what's sometimes called the unknown unknowns. With the less sensitive techniques, in fact, we can measure the known unknowns. So in NMR, we're sort of in a safe space. And things that we're detecting are things that have already been characterized and therefore can be explainable and understood. If many of the things that we're identifying are simply postulated metabolites, it's very hard to publish. It's very hard to develop biomarkers. So this area of the unknown unknowns represents a key area that has to be explored much more extensively, and some of the things that are, I think, coming out of CHEER and other efforts through the NIH and NIHS are helping in that. There are also two different routes to metabolomics. One is called an untargeted or profiling method, or simply chemometric. In this case, the spectral data is looked upon as more a pattern, a collection of peaks. And we compare the peaks and group the peaks using multivariate statistics. This is an example of a PCA, or principal components analysis plot. The other approach to metabolomics is a quantitative or targeted approach. In this case, rather than looking at the spectrum as a collection of peaks, you try and look at the spectrum as a collection of compounds, and you assign every peak, you quantify every peak, and you have authentic standards to make sure that you have those peaks. This approach was historically the more common approach in metabolomics. This one's becoming more widespread, particularly as metabolomics moves into the field of medicine, where absolute identification and absolute quantification are critical. Now, one of the central problems, and I brought this up before, with metabolomics was that we didn't have the tools historically to identify those peaks, to do peak assignment. Whereas in genomics and proteomics, we've had these tools for a long time. So we've had things like BLAST to do sequence searches. So sequence an unknown gene, blast it. You can usually kind of get an idea what it's like. Proteomics, run a 2D gel or an HPLC, you get some mass spec, send it to Mascot, and you can figure out what the proteins are. Metabolomics, for the longest time, you had a nice spectrum, sent your spectrum somewhere. Well, there wasn't anywhere to go, and we couldn't get metabolite identifiers or concentrations. So that was the kind of the state of the art or problem back in 2005. And I got involved in metabolomics a little bit before, but more actively beginning in 2005, where we worked on the Human Metabolome Project. So I think everyone's heard of the Human Genome Project. Probably no one's heard of the Human Metabolome Project, but it was a Canadian-based effort, which is really to do exactly what we did with the genome, which is to characterize all of the metabolites in the human body, to provide that information, to provide concentration data using a range of experimental techniques, but also to mine the existing literature. And we wanted to look at biofluids like blood and urine and CSF, but also tissues. So we worked on this for about, well, it's still ongoing, but we had funding for about 5 years. And we've completed a good chunk of that and have compiled a lot of it and made it all freely available, just as people have done with the human genome. So these are some examples of some of the papers that we've published looking at the human serum metabolome, the human urine metabolome. We've just submitted one on the saliva metabolome. We've done one on the cerebral spinal fluid metabolome. And in these cases, we're finding thousands of metabolites and producing concentrations for many of them. And a lot of that information was used in the paper that Steve Rappaport talked about in the blood exposome. When we started on this project in 2005, we just thought there was just one metabolome, but we quickly realized, in fact, there are many metabolomes because humans eat other metabolomes. So there are the endogenous compounds that our body produces and processes, and they range from femtomolar to high millimolar concentrations. There's about 20,000 endogenous ones. But people take drugs, so those also show up, so the drug-ome, if you want. There's 1,500, almost 1,600 that we've cataloged. We certainly haven't found them in one individual, but over populations you will find those. They're the foods we eat, and the food additives and the phytochemicals, approaching about 30,000. There are the drug metabolites that are processed through the microbiome and the liver. And then there are the environmental exposures, the pollutants, the things that you would hope would be at lower or much lower concentrations. And this graph largely reflects the finding that Steve mentioned before in terms of the scale or distribution of these components. What I've also indicated is the location of these different repositories. So the human metabolome database is where we've put most of the endogenous metabolites. Drug bank is where we've put information on most of the drug metabolites. FoodDB is where we keep all the data on the food components. And the toxic exposome database, or T3DB, is where we keep the data on the environmental and toxic exposures. So we've separated these out into different databases because they appeal to different groups, essentially, and for different purposes. All of this, as I said, is online. We maintain these websites at the University of Alberta and are updating them almost daily. They're widely used. Drug Bank gets about 10 million hits a year. HMDB about 4 million hits a year. They're not just lists of metabolites or drugs, they're very comprehensive. So they include all of the targets and receptors and have descriptions about them and lots of chemical information and lots of spectral information. And so people use these things for a whole variety of purposes, not only just for science, but also just to understand and explain things. So lots of them are used in education and training as well. And all of the drug companies use Drug Bank for helping with drug target identification. So the Human Metabolome Database right now contains almost 42,000 compounds. We have classified them into quantified and detected, as well as expected ones, which are ones that we know from biology and physiology that they have to exist, but we just haven't got the sensitivity to detect them. We've collected lots of concentration data. We've linked them to many, many diseases, lots of reference spectra. And the tools are designed so people can use them to search not only just by text, but they can search by spectra, they can search by structure, they can search by sequence, and so on. All of the data is free and freely downloadable. Drug Bank is another resource that we've compiled, which again allows us to look into both the drugs and drug metabolites, and lots of information about the metabolic processes, the transport processes, lots of spectral data, lots of drug targets and drug-drug interactions. Again, it appeals to a different group, typically, more pharmacologists than exposure scientists, but it is part of the exposome, and I think it's an important one. The one that's probably of greatest interest to people here is what we call T3DB, or the Toxic Exposome Database. And this is where we've tried to compile the toxic compounds. Some of them are the toxic drugs. Not every drug is necessarily toxic, I guess, but at any given dose. Obviously, things like pesticides, herbicides, endocrine disruptors, carcinogens, all of those have been collected. The mechanisms, binding constant, target information has also been collected, and the gene chemical links have also been compiled. So it's a very extensive database. It too also has spectral data to enable things like metabolomics. So, as I said, the initial problem with metabolomics is that we didn't have those tools that allowed us to take a spectrum, submit it, and get an answer. And thanks to some of the work we've done, but also work done by many other groups, some of whom are here, we now have those tools. And so this is, the blast, if you want, for metabolomics, or the mascot for metabolomics. These are now becoming available. And so that should enable much faster, much higher throughput, much cheaper metabolomic characterization. So in terms of what is possible with metabolomics, this is sort of an illustration of the tools that we use in my lab, but it obviously would differ with different equipment in different labs. So with ICP-MS, looking at metals, it's possible we routinely measure about 50 metals using small volumes, about 300 microliters. We can get down to picomole sensitivity. With NMR-based metabolomics, we typically measure about 150 compounds. We can work with about 300 microliters. Typically, it's micromolar sensitivity. GCMS metabolomics, 150, maybe 200 compounds, depending on your instruments. Again, sub-micromolar sensitivity, much smaller volumes. MS, or MS, direct injection, or LCMS, it's possible to identify and quantify anywhere from 180 to about 400 compounds routinely. With untargeted or relative quantitation, people are often getting up to 2,000 or 3,000 compounds with as little as 10 microliters. And then lipidomics, which is a branch of metabolomics, is one where you can usually hit a big jackpot where you can measure thousands of lipids. And that's because there are so many different types of lipids. So that's typically what's possible. So that's a lot of compounds that can be measured through metabolomics. Some of them are endogenous, some of them are exogenous, but it's obviously a lot of data that can be routinely measured. To measure, or when you're dealing with so many pieces of data, you have to use a lot of computer power. We'll talk about this, or we'll hear more about it later on, but this just illustrates the type of tools that we typically use in metabolomic data processing. So once we have our list of compounds, our list of peaks, we'll do things like Principal component analysis, or partial least squares discriminant analysis, PLSDA. We'll look at variable importance plots, identifying which are the most important metabolites, looking at their VIP values. So something more than 2 or 1.5 is usually important. We use heat maps, just like people in the microwave world. We calculate ROC curves to measure sensitivity and specificity for the biomarkers that we measure. We do a lot of pathway analysis. And I think, as Steve brought up, many of the metabolites that we look at in terms of pathways aren't simply to assemble or disassemble food components. Most metabolites actually have a signaling role. And this has been grossly neglected for decades. And it's because they're at such high concentrations, we don't often look at them as being signaling molecules. So whether it's the statistics, the pathway analysis, or just some cases the literature or database searches, all of these are used to help inform us of what we've discovered or what we're finding. Just to close off in the last few minutes, I wanted to talk a little bit about some of the emerging technology trends. Again, we're going to hear about this later on, but as a way of segueing into the next presentations. So we've talked about how do we speed up metabolomics. There are now trends in automated metabolomics. There are increasing efforts to look at disease and exposure biomarkers, and a number of them are being identified and published. There's exciting developments in tissue imaging or chemical imaging that are based on mass spectrometry and metabolomics. There's significant efforts to miniaturize metabolomic devices. Some of you might have heard of these tricorder concepts, and these are starting to move on. And then Addictomics, something that Steve Rappaport has been pioneering, is another, I think, emerging trend. So I'll just quickly go through. As I said, automation is critical. So if it takes us days to measure thousands using the manual approaches we currently use, can we automate that? And the trends are happening. Companies are doing this now. Labs are developing this. It's around the corner, or it's already here. So this is an important step and advancement in the field of metabolomics. In terms of biomarkers, obviously we think of diagnostic and prognostic biomarkers. The predictive ones are perhaps the more exciting ones, but what we're concerned about here are the markers of effect and the markers of exposure, or Steve, I think, was calling the markers of reactivity and markers of exposure. The ones in red are the ones that we're probably most interested when it comes to the exposure studies and exposome. These are some examples of biomarkers that my lab has been characterizing and identifying, and these are all examples of conditions that have an environmental cause, and where some interesting biomarkers have emerged, some of which are internal, some of which are external. Many may be related to diet, others perhaps to the microflora, others related to gas exposure, in one case with the fetal growth restriction. So these are examples, and these are measuring the areas under the curve, and we're using relatively small numbers of metabolites, but we're getting ROC curves that are actually quite astonishing. Areas under the curve of 90%. And that essentially underlines the power of metabolomics and metabolite measurement to be able to identify, diagnose, or even predict some of these conditions based on exposures or the metabolites that we measure. Metabolite imaging using MALDI-MS, I think, is an exciting development, and it's advancing very quickly, moving towards matrix-free MALDI, which then allows you to measure metabolites at very low masses, essentially spanning the full range of the metabolome. The techniques are getting better, faster, resolution is getting better as well, and I think these are exciting times, which will allow us to do a lot of tissue mapping that was unimaginable just a few years ago. The miniaturization process, on one hand, is the large NMRs that we sometimes use for metabolomics, not something you can carry on your back. You can obviously do something you can carry on your hand, and there are exactly some testing devices. The iStat is one that's actually used in the clinic, but there are others that are starting to roll out that measure 10, 20, 30 metabolites at a time. The glucose meter is perhaps the world's most widely used metabolomic device, and that's a handheld metabolomic device. Addictomics, and this is something that bridges both metabolomics and proteomics, and it allows us to look at exposures, the adducts, the modifications, the reactive electrophiles that bind to or modify proteins. And Steve has pioneered a lot of this, where we're looking at serum albumin adducts, and these can be characterized by mass spec. So you look at the protein, but break it up to look at the metabolites. And this again reveals some very, very useful data about what people are exposed to over long periods of time. And this is one of the challenges in metabolomics, is to be able to look backwards more than just a few hours. We want to be able to look back months or potentially even years. And so that means we're going to have to think about sampling new types of tissue, like hair or fingernails or bone or proteins. So metabolomics, I think the message here is relevant to exposure science, and what we're going to hear more about in the next day is really about how metabolomics and the techniques associated with can really enable exposure science. It allows us to do comprehensive quantitative compound analysis from very complex mixtures, not just pure sets. So that's the game changer. It allows us to do quantitative phenotyping. That's a game changer. It's allowing us to identify causes and consequences of environmental exposure. And that's the EWAS component that Steve was bringing up. With automation, with improvements in sensitivity, with the databases that I've talked about and the software that's emerging, it can be fast, it's getting cheaper, it's obviously very informative. And I think what you'll hear for the rest of today is that it's rapidly evolving. It's in the early days still. And monthly we're seeing, I think, substantial changes and advancements in the field. So these are exciting times. So with that, I'll just thank some of my collaborators and our funders, and thank you for listening.